Hello everyone. I hope you're having a good day. Really excited to be with you here as part of this virtual MathBio Rare Disease Day event. My name is Morten Søgaard of Astellas Gene Therapies. I'm really excited to be here today to tell you a little bit about our efforts on rare disease research and in particular, what we are doing on gene therapy. So, of course, really important for us to think about why we are here today, and I'm sure I'm speaking for everyone here that we are inspired by the patients and their sufferings and terrible diseases that are often cutting their lives in short. And that's certainly one of the reasons I and everyone at Astellas Gene Therapies are getting up every morning. That inspiration of the patients and the families. At the same time, also here, it kind of gives us a great responsibility. Being in this industry, we have so much opportunity to really change the lives and sometimes actually save the lives of these patients. And so I think that's really important for us, of course, to keep in mind and do good on that opportunity. So before we jump into our presentation, I just wanted to mention on the previous slide that was one of our patients, for our Pompe gene therapy treatment that we currently have in clinical trials as an investigational therapy. So Pompe disease, as you might be aware, is a severe progressive congenital neuromuscular disease. The overall incidence is estimated to be approximately 1 in 40,000 births, although frequency and disease progression varies with age of onset, ethnicity, and geography. The disease is caused by mutations in the gene encoding an enzyme called acid alpha-glucosidase. And if you have two defective copies of this particular gene, it means that you can't degrade glycogen in the lysosome. And the main effect of that is that you accumulate glycogen, and that leads to, pathology, particularly in skeletal muscle, which is really what is driving the disease. There are really no good therapies or adequate therapies available today. There's been kind of enzyme replacement therapy or ERT, where you basically give infusion of the recombinant protein biweekly, typically. And the challenge with that therapy is basically two challenges. One is that you would typically kind of generate antibodies to the recombinant alpha-glucosidase, and that is because most patients they don't produce a full-length enzyme, and so it's recognized as a foreign protein when it's kind of coming into the body, and that of course severely limits the kind of efficiency of the therapy because of the antibodies to the enzyme. The other challenge is that it's not kind of efficiently penetrating into skeletal muscle, which is really where we have the disease. So we're really excited at Astellas Gene Therapies, where we have developed an investigational, therapy for Pompe disease, called AC845. Which actually specifically targets skeletal muscles. So we have great hopes for that, and I'm really excited. And I think that's one example of how Astellas, is really pushing the boundaries of what is possible to, discover, develop, and deliver, sense of, first of their kind therapies, and particularly, of course, in this case for rare disease patients, really with life-changing values, and of course. One of the main reasons or really the reason that Astellas decided to invest in therapy, because we have such an important mission, with the rare disease patients. So, why do we work in gene therapy? And that's really because, as I mentioned, before that gene therapy has the potential to be transformative for patients and families, and really addressing by addressing the root cause of the disease. But at SELLAS, we have this focus area approach, to our drug discovery. We have actually 5 of these primary focus areas. One of them is focused on gene therapy, and we like to think about, opportunity here as this triangle where we need to kind of thoroughly understand the disease, what's the natural history, progression of the disease, biomarkers, etiology, and then importantly also really understand the underlying biology, mechanism of action, what is the pathophysiology, because that's of course important to understand in terms of how can we actually modulate the disease and what kind of modality can we apply, and of course for gene therapy we would typically apply kind of AAV-based gene therapy, and for gene therapy this triangle is particularly strong with a very strong confidence and rationale, and of course that's because we understand the disease very well. It's caused by a specific mutation, and we then use a treatment that specifically addresses that kind of mutation. So much better link to disease modification and best case cure than you have with most other diseases. And so I think I wanted to now switch gears, talk a little bit about SELLAS's commitment to gene therapy. I've just been at the company for 6 months, really inspired about that bold mission of the company. Most of you are probably not aware, but SELLAS made a very bold decision a couple of years ago to basically focus most of with R&D dollars on gene therapy and cell therapy, and specifically gene therapy, to address the terrible diseases kind of suffered by rare disease patients, and so that I would say is the main driver for that decision. So SELLAS really has made an investment in gene therapy that's on par even with much larger companies, just because we really have doubled down on these really emerging, but I think also transformative kind of modalities, and so we're really excited about the opportunities that we have in gene therapy, and of course also aware of our obligations to really move forward rapidly and bring medicines forward to patients. Really, the history here was the acquisition of Audentes, a biotech company based in South San Francisco. This last year, became SELLAS Gene Therapies as a gene therapy center of excellence, and then we've been this year actually significantly ramping up our investment in research, and also we built a new manufacturing facility in Sanford, North Carolina, that also bring medicines to market for commercial kind of use. So just very briefly here, history of gene therapy, just where we have gone in the last actually now close to 5 decades. The idea of gene therapy came up really at the same time as recombinant DNA kind of technology emerged, actually also in South San Francisco, and of course it was immediately clear, or that vision came up, could we actually address these terrible genetic diseases and rare disease patients? And fast forward a couple of decades to the 90s, there were the first uses of adenocyte-associated virus, or AAV, for gene therapy. Unfortunately, also some very terrible cases here of moving therapies too quickly into to patients, without carefully considering side effects and such, and understanding the biology. And the last couple of decades has been used to better understand the therapeutic modality and addressing some of those challenges. So now that is largely safe treatment here for in vivo gene therapy. We are starting to see several approved medicines here in the last part of the last decade, and then also we're starting to see some some ex vivo therapy as well, such as CAR-T therapy. But in this case, 2 approved medicines for some very severe rare diseases, retinitis pigmentosa and SMA. So, how do we address the disease and gene therapy? We can do that basically in 3 different ways. We can basically do what's called gene transfer replacement, and that's basically using the AAV vector typically by expressing a healthy version of the mutated gene, and thereby, ameliorating or treating the disease. And this is basically what we are doing in our ATA45 on the rare disease program that we just told you about. Another approach is to use gene editing, that's using CRISPR and, related nucleases. Gave you Nobel Prize a couple of years ago, you probably recall. And you can do a couple of things. You can do like a single cut. You can basically do a frame shift mutation in a gene. So basically, you would abrogate expression of a particular transcript, or in a little bit more technically complicated process, you can actually also repair the mutation. So gene editing is typically used for genetic diseases that are so-called gain of function, where you're expressing a toxic transcript of a protein. And then you have transcriptional regulation, where you typically would bind a protein to a promoter region of the gene where you want to regulate the transcript, and then you choose a repressor or transcriptional activator to that particular binding protein, or you can use shRNA or siRNA to knock down the transcription. So for AAV-based gene therapy that we are primarily using for rare disease patients is the gene replacement and transcriptional regulation are the main approaches that they use. For gene editing, you have to be careful with lifelong or prolonged expression, because these are bacterial proteins, and you have a risk of immune response and SAEs, if you express this in SAE. But technology is evolving, and maybe something we learn how to do safely. And then the question here is that how do we deliver these kind of gene therapies? Basically 2 main approaches. That's the in vivo gene therapy, which is typically AAV vectors, and which are largely safe to administer, and of course depends case by case on the disease, but have an attractive safety profile. So that's used typically for in vivo therapy, and that is what we're using at Astellas Gene Therapy to basically create novel breakthrough medicine for rare disease patients. You can also do an ex vivo approach, where you would take cells out and you would modify them, typically with the lentivirus-based approach, and you can create novel medicines such as CAR-T therapies or hematological diseases such as sickle cell disease. So that's also a very promising area that Astellas is investing in, side by side with our AAV gene therapies for rare disease. Okay, so based on that very substantial investment that we have made at Astellas in gene therapies, we have one of the broadest early pipelines in the industry, and a lot of that pipeline was created through acquisitions such as the one we did at Potentis. We're also doing partnerships. You might have seen a recent partnership with Taysha to kind of acquire an option to 2 of their kind of clinical stage or preclinical stage programs, and we're certainly looking also at other opportunities to kind of strengthen our pipelines and bring novel gene therapies to rare disease patients faster. And in addition to that, we are also investing heavily internally, and have a very strong internal research, top-notch research engine to bring novel differentiated therapies forward. And as we do that, of course, we also, carefully consider how can we bring the most, recent, kind of technologies on board to really make sure that that we have the, the most competitive therapies possible. And investment, really covers capabilities across the value chain, from research to validation, process development, manufacturing, clinical trial operations, and of course we have to make sure that we bring medicines forward that are safe and also well coordinated with the regulatory authorities, so that we can bring the medicine, to patients as quickly and as safely as possible. So currently, and I'll just give a little bit of an insight into our footprint here. Most of our research actually is based in South San Francisco, and that came out of the Audentes acquisition a couple of years ago. We also have a small team in Tsukuba, Japan, which is the main research center in Japan for Astellas research. And then we have GMP manufacturing in South San Francisco, and also, as I mentioned before, we kind of built a large facility in North Carolina that will allow us to bring commercial products forward, also for neuromuscular diseases, and which require lots of material. So we're really gearing up, both on the research front as well as on the manufacturing front to really, bring forward novel therapies for gene therapies for ASD patients. So as mentioned here, just like a few of our current programs that are currently disclosed, but we arguably have one of the broadest pipelines in the industry. So we have basically two clinical programs, AT845 for Pompe disease, that I mentioned earlier. We have AT132 for X-linked myotubular myopathy. It's a terrible disease, with great promise. And the therapy is a very, vulnerable patient population. So it's a treatment that has its challenges, but we are very committed and excited about the opportunities here. So neuromuscular disease has been a great area of great focus, and then beyond neuromuscular disease, we are also, focused on ocular disease, which is an area of traditional, strong strength for Astellas, and also we are moving increasingly to CNS disorders. And just one example here for Friedreich's ataxia, we'll be looking at potentially treating the CNS aspects of that disease, but we have also moving forward as an investigational therapy, not yet in the clinic, but hopefully there soon on kind of the cardiac targeted aspects of Friedreich's ataxia, and that's important because most of the Friedreich patients, they die from cardiac complications. So very important for patients. So we're really excited about the opportunities that these new medicines, transformational medicines, or investigational medicines, of course not yet in the marketplace, but we are moving them forward as quickly as we can, and a lot of exciting opportunities here that we're excited about every day when we go to work. Just here at the end, just wanted to talk a little bit about Astellas Innovation Engagement Initiatives. Astellas has a very significant presence in the U.S., mostly through biotech acquisition. So in the biotech, in the Boston area, and then also in San Francisco and Seattle, like our main areas of presence in terms of R&D. And we also are very active participant in the local, kind of innovation ecosystem. So we are, kind of the founding sponsor of Lab Central 238 in Cambridge, focused on cell therapy and gene therapy. Very active on Lab Central 700, 610 as well, and we have a significant presence, senior role for the MPT biolabs in the Bay Area. So I just wanted to maybe stop there and thank you all for your time, and also MassBio for the invitation and the opportunity to discuss our plans and visions for rare disease and where we hope we can really provide important contributions and new medicines to rare disease patients. Thank you very much.
That was a great talk by Morten Søgaard from Astellas. Hello, everyone, and welcome to Cardiovascular, Metabolic, and Rare Disease Day of Innovation Week. My name is Gauri Nair, and I'm the Vice President of Innovation at MassBio. We are happy to have the opportunity to connect with you today. If you have any questions for Morten, please put them in the chat window. In the meantime, Morten, I'm going to ask you a few questions that have come in. What a great talk. What is Astellas looking for in terms of gene therapy partnerships?
Thank you so much, Gauri. And first of all, thank you for the invitation. It's really, pleasure and a privilege, to be able to give this talk here at in MassBio Innovation Week. Well, I would say we're looking in a couple of different areas, right? We're certainly looking at strengthening our portfolio. So in terms of, I would say, clinical stage assets or kind of R&D stage assets. You might have seen some recent news on partnerships we made, and so that's definitely an area of interest. And then gene therapy, of course, we have to make sure that we stay up to stay competitive in terms of new technologies. So things like novel capsids, like pre-existing immunity, cell production technologies and such, and those are all areas of interest to us. And then we're also looking for partnerships with academics as well as with early stage biotech companies for accessing the latest science and also academic experience and expertise.
That's fantastic. So what does a strong partnership look like for Astellas?
Yeah, that's a good question. Well, first of all, I think you need to be strategically aligned, right? I mean, so it needs to be, and so I think you need to be very thoughtful, also not, dabbling around, really think about, what is it that's really, what's going to strengthen your, internal portfolio, but also then external assets that you're looking at. So you just have to be very mindful on that. And then of course, it's very important that you're aligned also in terms of culture and chemistry. And we very much like, collaborations that are all in, or, where you have an open, close and trustful relationship, as is in any, relationship in life, also in partnerships here on the business side, it's really important.
Okay. Do you have any grants for ultra rare disease societies? Does Astellas provide grants?
Oh, grants for ultra rare diseases. That's a good question. I'm relatively new to the company. I've been here 6 months, so I'm not aware that we do, but it's a very good question, and I will look into that after this presentation.
Yeah. Another question that came in was, what challenges do you see for gene editing and therapies in that space? What are the biggest challenges?
Oh, for gene editing specifically?
Yeah.
That's a good question. I think gene editing is really, of course, promising technology. I mean, we had a Nobel Prize a few years ago, and then we had, tremendous progress on kind of basically base editing, more like prime editing and similar technologies. So I think a lot of potential I think certainly for mRNA LNP delivery, I think there are a number of companies, that have really promising products in the pipeline, in particular targeting the liver, and I think good cost of goods for mRNA LNP and relatively good safety profile. So I think that's an exciting space. Of course, for mRNA LNP, the question is delivery. Delivery is often the challenge. How do you get it to the right place? If we think specifically for AAV gene therapy, I think here, of course, we have to be mindful of expressing a bacterial, kind of protease longer term. And of course, there have been some recent, very, sad developments that I think most of you have probably followed. So I think this is something we have to be very careful with safety. So I think for AAV delivery, I think it's quite challenging. But of course, if we can manage it technically and safety-wise, it's very attractive. Of course, if you can actually repair the mutation, and also you can address gain-of-function mutations, genetic disease. I think this is where it's particularly promising, I think.
Yeah. So we recently concluded our year's cohorts for the Accelerator MassBio Drive that we run for early stage startups. And we had one of our companies, actually HybridX from Yale. It's a spin-out that's working on safe gene editing.
Oh.
And that's kind of trying to address one of these issues that we see in gene therapy.
Yeah, maybe if you could connect me afterwards, I would love to learn more about it. Yeah,
absolutely, we'd be happy to do that. So one of the other pieces, what would be a good advice you could give to gene therapy innovators, young startups that are coming out in this space?
Yeah, no, good question. Well, certainly, at the core of innovation, you have to have a good understanding of, how does your innovation actually fit in to the overall landscape, and what is the value proposition versus other technologies out there. Was it, are you focused on a specific asset, is a specific technology? And I think so really having done good homework there in terms of positioning versus competition, I think that's important. Then certainly looking into like IP situation, making sure you're well positioned there. Then of course, getting a strong team in place, of course, it's kind of like the vanilla kind of advice, right? Strong management team, that's also what investors are looking for. And then of course, getting a couple of, well reputable, venture groups, if you're looking for venture capital, certainly I think would be strengthening the proposition. I would say don't forget about pharma. There's the venture groups and pharma that are often well aligned with the strategic interest of the company. So I think that could also be an opportunity for the kind of innovators. And then I would say resilience. This is a really exciting area, exciting time for gene therapy. And I think if we dial out, like 10 years, I think we'll all be amazed to see what will have been accomplished in the last 10 years. So keep up, the hope and the spirit and resilience. That's I think really important.
Well, thank you, Martin. That was wonderful. And we are just out of time for the Q&A, so we'll be moving on to our next speaker.
Yeah. Thank you so much. My pleasure.
Martin.
Yeah.
Thank you for your time.
Yeah. Oh, my pleasure. Yeah, thank you again to MassBio. Yeah. Thanks.
Glad you could join us today.
Yeah, thank you.
It is my pleasure to introduce our next speaker from Sanofi, Raquel Mura. She is the Vice President and the Global Head of R&D Operations for North America at Sanofi. Thank you.
Thank you, Gauri. It's a pleasure to be here today. I'm very proud, together with my Sanofi colleagues, to be here on what is a first of its kind event where we have this opportunity to convene and celebrate diverse entrepreneurs, those of you who typically come from underrepresented backgrounds in our industry, but that are also working towards greater health equity so that we can continue to deliver for the communities that we serve. Today, I would like to speak briefly about two things. So the first is reiterate our commitment at Sanofi and our heritage when it comes to advanced scientific discovery for patients with rare diseases. But I also wanted to talk a little bit more broadly about structural issues related to DE&I. I hope at the end of my talk that you will join me in my call to action so that we can continue to advance the agenda of DE&I across our industry and the sector. So speaking a little bit about rare diseases and why is it so special for Sanofi. First of all, it's a very challenging field, as many of you are aware, where innovation is highly critical to advance medicine for our patients. Those patients and their families, they can endure a long healthcare journey where there are tests involved, misdiagnoses, and consultations with specialty doctors. It's a very tortuous journey, and we are highly dedicated to shortening this journey by delivering transformative healthcare options. In the United States today, we have about 25 million Americans living with a rare disease, but then more than 350 million people around the globe are affected by those diseases. At Sanofi, we have a heritage of over past 40 years to advance the field for rare disease patients. We have established our leadership back in 1984 with a successful treatment for Gaucher disease. And of course, we have also contributed to the longstanding stewardship of the multinational disease registers because that is so important for us to create a strong center of gravity for those patients' communities since they are so sparsely distributed throughout the world. As far as scientific discoveries and transformative medicines being brought up in the market, I'd like to highlight some of the recent examples of successes, including Zempozyme, which is the first disease-specific treatment for ASMD. It's an extremely rare disease diagnosed with less than 120 patients in the USA that did not have, up to this point, an effective treatment. So there are many serious life-threatening diseases with young patients passing away before the age of 3. And many patients could not access ours or other clinical trials because of safety reasons. And because of innovation, those patients now have a therapeutic option. But part of this innovation comes from the diverse pool of talents and experts that have been engaged to be able to make this scientific discovery. Which brings me to my second point, which is how might we, as an industry, as a sector, as a society, continue to foster diversity so that innovation can be more inclusive. And we all have a role to play. There are a few structural issues, though, that we must first talk about and then address. We know the scientific discovery is richer when it's equitable and inclusive. Also because it allows us to bring all the different perspectives and richness of experiences from all those that are involved. Specifically in our industry, but in others, what we know is that by having more diversity and inclusion among the teams and among those that are involved in those discoveries, we are able to bring richness of ideas, but not only that, experiences of perspectives, different experiences and perspectives. And as a result, being able to bring more meaningful solutions to patients. We also know data from McKinsey that the most diverse companies are more likely to outperform their peers in profitability. So we not only have the societal business case, but we have an actual business case for diversity. We believe that leadership is important when it comes to diversity and inclusion, and not only senior leaders and executive committees play a role, but leaders across the different functions as well. We know specifically when it comes to underserved and underrepresented minorities that access to education can actually lead to economic advancement, and particularly when it comes to women. Data from the United Nations shows that where women are empowered and are able to advance, actually society prospers. I'm going to share a few examples of what we are doing at our own company, Sanofi, and some of those examples are also being shown and exemplified in other companies in the industry. And again, I hope many of you would join me in my call to action on how we can drive industry-wide, sector-wide transformation when it comes to diversity and inclusion. First of all, workplaces, they must reflect the society that we live in. And that means making sure that we are not only demonstrating inclusive behaviors, but that we are giving opportunities for those communities where we do business to bring in talents from different backgrounds. It is so important that our employers and those partners and suppliers whom we engage are representative not only of the people who use our therapies, but are representative of the communities where we do business. With that, we're able to strengthen the economic development for those communities and then contribute for greater health equity. Speaking a little bit about talent now, how might we, as an industry, as companies, make the case and internally and take action to drive diversity and inclusion? So the first thing, and it's been an area of focus for us, is to ensure that we are nurturing diverse talent. So from our perspective, we've been spending quite a bit of time and effort partnering with university and community colleges and schools as well, in some cases even at high school, to make sure we're not only engaging students to share more about STEM education, but also provide opportunities for them to get firsthand experience. That means hiring interns and trainees that come from diverse backgrounds, but also nurture them as they come and join our organization to understand what a career in STEM could look like. We have some flagship programs, which I won't go through here today, but it's so important once we go through this and we bring the diverse talent into our own organization that they feel included in many of the activities, and most importantly, the culture that we have, so that they can have a purposeful experience. And that includes grassroots initiatives. For example, ERGs, many companies have employee resource groups that allow those talents that are coming from different backgrounds to feel a bit more immersed into the culture of the organization, but also to be able to find similar talent with whom they can talk about issues, but also have open dialogue with leaders. And then mentoring and allyship programs. So once we have the talent, how can we make sure that we are providing them opportunities to be mentored by different leaders in the organization? We have actions also for managers, to name a few very briefly, ensuring that we are mindful of candidate slates, and if there is a diverse slate when it comes to talent. Are there any biases in the process? Are we engaging diverse interview panels when we bring in new talent into the organization? And again, when we look at considerations for promotions and any issues with retention, being mindful of any challenges there. Of course, promotions and hiring should always be based on merit, but being aware of potential bias is something very important as well. So how can we make sure that for patients, we are developing treatments that will ensure that we have access to medicines for everyone, including for some underserved communities where typically reach may not be there. And here I would like to highlight a program that we have recently launched with Howard University, where we are actually not only bringing diverse talent into the organization, but looking at a pathway where they can actually be hired. This is just one example, but the idea is that by trying to address some of those inequities in opportunities, we will not only help society prosper from a diversity, equity, and inclusion perspective, but also deliver better outcomes to all patients. Now, in terms of the call to action, and I would like to ask all of you to join me on my call to action. First of all, change is happening. It is happening, and we would like it to happen even faster in our industry. And I think we have several examples of companies that have been highly committed, and we are as well. But first of all, we need to think of those changes as structural issues that we need to continue to address. The first is for leaders in the industry, but all of us, to really understand the importance of the issue and role model. Ask our diverse colleagues, partners, and suppliers, how are things going? Is there anything we can do differently? And for those of us that are part of the company, continue to provide access to trainings, to development, and then ensure that we are having those honest conversations. And then talk a little bit about culture. What is the culture that we are creating in our organization? Are we creating a culture where diversity and inclusion and equity are actually valued on a day-to-day basis? Do colleagues and employees feel comfortable having two-way conversations, not only one-way conversations, for both, for those of that need to understand a little bit more how we can foster those opportunities, and for those that are coming in? Pave the way for meaningful and lasting changes by role modeling. That is so important. But also talk to teams. Are they feeling safe? Again, do people feel comfortable raising the difficult questions that come when diversity becomes such an important topic in those relationships? And then monitor for things like microaggressions. We know that in many cases, those take a toll, especially for diverse talent, in terms of the energy toll that it may take. We're very fortunate to have an environment today where we know from research and surveys that we have done where there is psychological safety, but it's equally important to drive change at the industry level that we are aware of that. For those of you that are pitching today for the entrepreneurs, first of all, I want to say that you matter. You are in a very unique position to drive change. I think we're all committed to make sure you have the opportunity, the visibility, and the exposure. And you're here because you do want to change things, and especially in the case of rare diseases, which is such an important area. But your impact will continue to be greater by all the leaders that are here helping you thrive. So we are all part of this change, and hopefully this is a long journey, but where we're making significant strides. Together, I am convinced that we can achieve a more diverse, equitable, and inclusive society, not only for all of us, but for the patients as well, and where scientific discovery and bringing transformative drugs to market can continue to take place. So with that, I want to thank you for your time today and wish you a great remainder of the session.
Thank you, Raquel. That was wonderful. I also want to thank Sanofi and Astellas for your wonderful partnership with MassBio. Both of you have been great pharma partners for us. You've contributed so much to the innovation ecosystem. You've always participated in all our events and supported us as we try to make it, as we try to support our members. Thank you so much.
Thank you, Gar. It's my pleasure.
Before we move on to our presentations, I want to make a few announcements. Please take a moment to look at all the sponsor booths on our virtual platform. Both Astellas and Sanofi have great company information. They have videos. Please do visit them along with all our other pharma and ecosystem sponsors. Please do put in your questions in the chat before the presentation end for all the startups coming up. Presentations will be approximately 20 minutes, and we'll be taking questions for 5 minutes after that. If we get them a little ahead of time, it's easy for us to, kind of ask the startup founders these questions. So please do put them before the presentation ends. I also want to tell you a little bit about some of the upcoming events we have that may be of interest to you. We have a makeshift forum tomorrow, December 8th. You can find details at massbio.org. This is going to be about religious diversity at work. My colleague Warner Santiago is hosting that. We also have a year in review panel, which is important for 2022, I think, because of the ups and downs we've seen this year. That's going to be next week. We have, again, a very accomplished panel of leaders from the Boston ecosystem. Omar Khalil from Sante Ventures, Wade Brown from DMS, Leslie Miller-Nicholson from MIT. They will all be speaking on this panel. Do look at our website and register for it if you're interested. And now, without much ado, I would like to introduce Reginald Swift from Rubix Life Science. I've had the pleasure of speaking to Reginald. I was introduced to him through our affinity partners, BeWise. And it's been a pleasure talking to you this fall. I'm so happy you can be here to present to our audience today. Thank you, Reggie.
All right. Thank you so much, Gauri. I certainly appreciate the time to be able to present in front of your crowd and being able to be, longstanding partners with MassBio and now just recently BeWise. So thank you again for the opportunity. So I know that my time ends at 1:30, so I'll definitely try to make this as efficient as possible. And let me share my screen for a second. All right. All right, can everybody see the screen now? All right, I am going to take that as a yes. So yes, thank you for being able to join me today. My name is Reginald Swift, Dr. Reginald Swift, founder, CEO of Rubix Life Sciences. And, to much what Raquel was saying from Sanofi around diversity and inclusion within not just healthcare access, but also just the continuum of clinical research. We tend to focus on that early discovery area where we want to make sure that data helps transform and translate. How do we target approaches where, clinical research can actually go to the patient, right? Can be able to be presided by and for those types of diverse communities, the ones that are hard to reach, the ones that are in low burden areas, the ones that are in low resource areas, the ones that are in austere environments where it could be hazardous, right? Because we want to make sure that when we think about medicine, we think about health, we think about just access totally. We don't have to, always have an asterisk to say, well, we have to focus on health, but also health equity, right? We want to be able to move the needle where health is just health, right? We're considering all demographics, all races, all backgrounds, no matter what type of platform we're creating, right? Whether it's what the industry is creating, whether it's infectious disease, whether it's rare disease, whether it's oncology, whether it's other chronic conditions, we're just focusing on that. So to continue, we started out in 2016. So we're a culturally competent contract research organization, and for Star Wars fans, C3RO instead of C3PO. So we're specializing in translating data and data analytics around those hard to reach patients in order for us to design clinical trials that are going to be adjacent in these communities, right? In these hard to reach communities. So we want to make sure that we are the change agents, that we're taking the information and the opportunity where whenever there's a new product, whenever there's a new specific clinical trial, we can design with those types of patients in mind, right? And our focus is to make sure that we become change agents, right? So where we are helping accelerate what Sanofi is doing, what Astellas is doing, what Pfizer, what Johnson & Johnson, Merck, and all the other pharma companies are doing. We're in several cities, primary locations here in Massachusetts, but we do have a lot of offices across the United States, where our makeup is about 71% women. Because we pride ourselves on making sure that we're diverse. We're making sure we're minority driven because we want to represent the community that we actually want to serve, especially when you think about clinical research and the sites that we want to create. We want to make sure that we have culturally competent individuals that can help us design ways in which we do outreach and being able to be, as a contract research organization, where we're fulfilling either phase 1 or phase 2 studies, we're accommodating as well. In our mission, we just want to make sure through that data, we're connecting and empowering the world's healthcare information systems, right, through diverse patient analytics, right? So it's being able to, you know, as I was saying earlier, translating those patient insights, those patient, gaps, right? Those patient data sets that we want to make sure that we design virtual, hybrid, or even traditional clinical sites where we can make sure that even though we may not have the opportunity to have a face-to-face with the patient every day, we're making sure that data is getting very much close to them up front and center so that we can learn about the patient, talk to the patient, and speak for the patient so that everything is becoming transparent where we're learning about them and they're learning about us at the same time. So that trust is built in. So since 1980, I want people to kind of think of this number, 4%. 4% of minorities have been involved in clinical trials since 1980, right? And over 225,000 of trials have been conducted since 1980. So that is a systemic problem that we've seen, time and time again. And even though it could be worse before that, and we know what the history is of the medical research, especially in the aspect of the United States. So we are no frown to it. We are no strangers to that, but we want to make sure that as we move forward in what the evolution of medicinal research is and therapeutic research is, we want to ensure that when we think about access, when we think about care, when we think about trials, when we think about data, right, we're inclusive of all demographics, no matter what the cost, right? Because the world is becoming much more diverse as we live and breathe every day. So we have to make sure that we take the effort and we make a sustainable process. And through what we want to be able to instill as a CRO, we want to do just that. All right. This is the way that we support our clients, whether it's through many of the programs that whether it's rare disease, infectious disease, but this is kind of the way that we think about the way we support data analytics. I know that word is kind of a big term, but the way that we think about it is making sure that it's patient healthcare metrics or analytics, right? To make sure that we understand from a multitude of variety of factors, whether it's access issues, whether it's bioenvironmental issues, because when we think about specific causes, because we're plugged in with the DOE and the EPA, where we're learning about patients in specific areas and toxicity in specific buildings and specific water and the treatment of specific water, right? And contaminants in specific water. There's a correlation between where they're living and the prognosis and prevalence of specific diseases. So we want to make sure that that type of information gets to individuals, companies like Sanofi, companies like Astellas, companies like J&J, Pfizer, and the big ones, and even the small ones as well. Because as we think about innovations, we have to think about where they are in the process of where patients live, right? To make sure that they can be eligible, to ensure that they can participate and still have that confidence that pharma is really taking care of them. Right. And then we're looking at specific predisposition genetic indicators that can help us really do high profile targeting in these communities, where then we're creating, as a targeted CRO, we're creating virtual sites, hybrid sites, even traditional sites, where we're going to be able to help manage throughout the life cycle, whether it's phase 1 and phase 2 for now, for those patients as well. So we do a lot of work with the federal government. Whether NIH, CDC, we've done a lot of work overseas with the CDC and WHO and the UN. We've launched about 19 different trials in infectious and rare disease and ultra rare disease. So with our federal clients, we've been able to do a lot of clinical trial design, a lot of clinical trial support, actually clinical trial initiation, engagement, and being able to kind of really take a look at how do we look at specific type of digital health tools, rather we can retrofit or fit for purpose need, where when we go into low burden, austere environments, how do we take a look at being able to utilize like a Nokia phone, the one you used to play Snake on, into environments where there is an infrastructure for miles, right? Or there isn't any type of semblance of first world and any capacity for miles, so that we look at ways on ensuring that we're using approaches that we are talking with the patient and to the patient and for the patient simultaneously, right? And as we're pulling that data, as we're creating systems and creating profiles and platforms, we're ensuring that that type of data, feeds back into what we're doing in the earlier sense, right? From business intelligence to advanced analytics to even patient engagement. It's helping us do that informed process so that we can be able to make sure that programs and initial programs that Sanofi will launch or anyone else will launch will take accommodation of to this data set that we can create and be able to provide. All right, and we pull a lot of data from a lot of places. We have many public-private partnerships, especially with the First Nations here in the United States. We are plugged into about 18 different First Nation tribes through IHS. And we have a lot of collection of data within CMS, as well as a lot of the federal institutions here as well. We are plugged into some of our laboratory and biorepository data partners that we provide on an ongoing basis for genetic coding, genetic profiling. And we certainly pull from local clinic and community hospital resources. So we're able to kind of continuously look at areas of concern of emergence of prevalence, right? The emergence of disease evolution, the emergence of how do we look at intimate building, because our first interaction was in that space, was in rare disease. So we know because the prevalence was a little bit harder to reach, we just helped design specific patient universes so that we can learn from the data that we're pulling on actually how do we look at the continuity of therapeutic discovery, right? And looking at how the progressing of patient natural effects will either matriculate or mature the disease, or they can mature their outcomes, right, for their own health. And much of our disease strengths are here. Again, much of our firepower has come in the form of what we've learned from the data sets that we've been able to pull from infectious disease, rare disease, and ultra rare. We are now kind of just expanding into oncology, but many of the things, like even to Rachel's point, Raquel's point, Gaucher disease is something that we also looked at as well, that we saw that there was a connection to Gaucher's and maternal PKU, as well as DIPG. So there are specific indicators that we were able to target specifically from either some nations of India as well as nations of Africa that we were able to kind of do some correlation studies with. So it's being able to learn in real time so that we can use real world evidence to be able to help really craft programs and assistance and development efforts, so that as we look at launching clinical trials, we're in these avenues where we are trying to be proactive, right? We want to make sure that the proactive nature for patients is ensuring that we are reaching out to them at the right time whenever they are going to be able to need some help and need some resources. This is kind of where we are in the world. We have about, I think there's a little bit more data than that, but we have been working worldwide. Total number of patient data sets is roughly almost 4 million, but we do have about close to 2 million patients fully consented to be able to provide their data for us, so that we can make sure that whenever there's a launch of a study, whenever we launch a study, or whenever a sponsor would like to work with us to support a study, we can be able to tap into these types of networks, whether in any of these demographics areas as well. So we're becoming more and more focused on these communities, and we want to continue along to be able to represent these communities, because when you think about where innovation has taken us, it's taken us in great places, because it really is, it's selling society, right, as far as medicine. But we can't forget about those that have not been able to have that access, right? We want to make sure that whenever we think about that 4% number, right, we want to be that Marine Corps type of company, that the Marine Corps of clinical research, so we can move that 4% up to 50%, right? So, and we know that it's going to take a collaborative effort across the industry and many other stakeholders to be able to do so. So we're optimistic about it, and we want to continue along that path and go from there. And from our genetic index, we can access quite a bit of biomarker data that helps us realize some of the avenues in where we want to kind of have high profile approaches for early indications for patients, so that we can make sure that as we receive like lab samples, we can be able to target these types of patients beforehand, so that they can have the education, right? They can have that knowledge. They can be able to be able to know that there could be boots on the ground to be able to support their need, or we can have a phone call with them, or we can send letters, we can send texts, we can send emails, we can send all types of assets to them at the right time, so that they can be able to be included in that environment, right? So I know that I'm coming up on my time for presenting, but would be happy to take any questions from the audience.
Thank you, Reggie. That was fantastic. Loved hearing about what Rubix is doing and how you're trying to, change things in your own way. I have a question for you. What inspired you to start Rubix, and what advice do you have for young startups in this space?
Well, thank you for the question. For me, it was a personal passion, more so because personally, my father passed away from cancer, right? Stage 4 cancer. And just knowing that there was a specific clinical trial for his condition and he wasn't able to access it, that was the tragedy, right? And for me, from a postmortem, I just always wanted to find out information as to how it happened, right? Find out information. And on the fact-finding mission, for me, I saw firsthand some of the gaps, some of the large gaps that are the glaring gaps that are there between not just the hospitals, but also community members, right? Also between the sponsors, also between the payers, also now we have PBMs, but between all of these groups, right? We want to be able to try to close those gaps, mitigate those challenges, so that people, like me, like you, like anyone else on this webinar and in the world today can feel safe that somebody's actually looking out for them, right? So for me, I love what I do. I really, I'm not here to be Superman. I'm just here to help people really get that opportunity, be able to not just be that fly on the wall, right? Not to be that wallflower, but actually become front and center in the eyes of pharma, right? So that anyone can be as important as they need to be when you think about clinical research. And I mean everyone.
I'm sorry to hear about your father. It's always hard to see a parent going through health challenges and end of life. I sympathize with you. I'm, in a similar situation, so do totally feel what you're saying.
Thank you.
One more question I have is, what are the challenges you've faced as you got this off the ground, and how can we as an ecosystem help you? How can MassBio help founders like you?
And I'm glad that you asked. So when I first started my initiative, my endeavor, it wasn't until, COVID-19 that kind of really illuminated the need for diversity, right? But I've been working on it since pre-COVID. And of course, there's other members in the industry that have been working on diversity for 20, 30 years. And we get that. And when I first started out, everyone was telling me, well, this isn't investable. This is not an area where we're concerned about. And everyone was just continuously focused on, the innovation, which, is important. I get it for diseases. But when you think about that saying, we're all patients, that has to be accommodated for a specific reason, because we are all patients, right? That has to make sure that we all can trust the medicine, no matter where you are. There is that disdain for pharma because we've always taken that same approach time and time again. That's why there's a bigger, that's why the trust factor for pharma has fallen since the 70s, right? And it was at an all-time high in the 70s. But we want to now kind of really just try to ramp that back up. And, for me, it's being able to understand that, even though the nature of pharma, the big pharma companies tend to kind of move a little bit slower than the startups, right, is to be able to have the bridges in which advocacy groups like MassBio can be able to start creating, just like how we have it here today, right, to make sure that there's topics that they can start thinking about where it doesn't have to be a firefight, where startups are trying to actually solve a really big problem, a real issue. And it, of course, validation has to occur of that problem and that issue and the technology and the solution. But also really just kind of have an open ear around listening to what the evolution of technology, the evolution of innovation is going to take us, right? Because I think whenever you think about 20 years, 30 years from now, you want to make sure that company is still going to be alive at that time, right? So, and that helps because we all know that small companies are much more agile and nimble to help take programs forward. So it's really just that opportunity for that bridge to kind of be strengthened and enriched.
Speaking of pharma, I think we heard at a DEI conference last July from Novartis, and they and other pharma companies have joined this initiative called Beacon of Hope to diversify clinical trials, and they're working with different affinity groups to make that happen. So this is so timely. Thank you. Absolutely. And one more final question before we move on to our next presenter. What's next for Rubix? What are you looking for in terms of partnerships with pharma? What's your ask out to the ecosystem?
Oh, well, my ask is, let's connect to look at what the data is going to be able to show us, right? Let's connect and partner to see how well we can move into the future, right, of trials and trial designs and hybridization of trials, right, and hybridization of clinical research. Because it's as you create agility within your teams and your functions and even the departments, right, it's going to be even more important, because the way that we have to reach diverse patients, we have to take nuanced approaches in order for us to get there, right? It can't just kind of be the same methodology, which we understand from the top. It has to look like that, but from a functional level, it can be evolved.
Thank you, Reggie. This was wonderful. I'm actually going to go on to our next presenter now, since we are up on time. Without much ado, I want to introduce Jonathan Su, who is the CTO of Gensake. I hope I'm saying the company name right. But Gensake was started right down the street at MIT, and it is hoping to reimagine genomic medicine and with their phage-derived particles for in vivo gene delivery. Did I get that right, Jonathan?
That sounds like a perfect description.
Thank you. Look forward to your presentation.
Yeah, first I'd like to thank the organizers at MassBio for giving us this platform to talk about our work. As mentioned, my name is Jonathan. I'm the CTO at Gensake, and we're really excited to share our story with you. So at Gensake, our mission is to ultimately develop redosable genomic medicines for patients with a variety of different genetic diseases. And how we plan to do so is by pioneering a new class of in vivo gene delivery vehicles, in which we refer to as genetically encoded nanoparticles, or GENPs. And what we believe these GENPs represent are nanoparticles that we see in nature that have intrinsic properties that potentially make a good gene delivery vehicle, such as cargo capacity, immune profile, manufacturing scale. And we essentially take those scaffolds and we evolve them to become more viral-like, to eventually efficiently work as a gene therapy. So the first member of this GENP class that we're looking into is a phage-derived particle, shown on the left. So the phage-derived particle has really interesting properties in that the field of phage display has shown that there are multiple engineering points on our capsid proteins to be able to functionalize in a multidimensional way on the capsid. In addition to that, inside this capsid, it naturally holds a large genetic DNA payload that exceeds 20 kilobases, which is well above the cargo capacity of current gene therapies. In addition, we believe the phage-derived particle has advantages when it comes to immune tolerance, in addition to manufacturing at scale, because we do this in a bacterial culture. The opportunity for our technology, we believe, is significant, with the overall vision of delivering various therapeutic cargoes to diverse tissue types in the human body. So our team, Avi Uyangan and I, we first met at MIT back in 2019, and we co-founded the company shortly after, with the helps of our scientific advisory board and professors Angela Belcher and Amin Hajitu at MIT and Imperial College, respectively. They are both pioneers in phage engineering and in vivo gene delivery. In addition to them, we have also taken on Dale Irvine from MIT to help us think through the immune aspects of our delivery platform. Our board of directors are headed by Jeremy Levine and Briggs Morrison, who are currently CEOs of public biotechnology companies and have led the development of many therapeutics in the industry. Our broad team consists of many leading KOLs and indications that we're interested in addition to a very diverse group of scientists that span molecular biologists, phage engineers, and tissue engineers. How we like to start us, okay, my next slide. A snapshot of our company is that we're currently based at Lab Central, where we have our 8 core team members in place. We recently raised a $5 million seed round from Ovid Therapeutics. On the IP front, we've exclusively licensed 6 patents from Imperial and MIT that really protect our foundational platform technology. In addition to that, over the course of the past year, we've filed 10 additional patents internally and have 5 others in the process. In terms of where we are in platform, we've signed 1 non-dilutive collaboration agreement with Ovid Therapeutics and have a couple others that are in due diligence. And from a technology point of view, we really have the platform up and running, and I'm excited to be able to talk a little bit that later on in this meeting. So when we think about the technology development at Genentech, we begin with the journey of DNA delivery. So we believe that there are 2 significant in vivo barriers that we need to overcome to be able to have successful DNA expression. So the first barrier is tissue-specific targeting. So we have to develop a vehicle that primarily engages with our on-target tissue of interest. In this schematic, it may be the lung, and not engage with off-target tissues such as the liver. And what we've seen with other delivery vehicles, such as adeno-associated virus or AAV and LNPs, is they have intrinsic natural tropisms to the liver, which has really limited their therapeutic window for targeting other types of organs. In addition to that barrier, we see the second barrier in intracellular trafficking, or what we refer to as the last mile of gene delivery, where DNA payloads need to be delivered into the nucleus cells to be a productive molecule. And whereas AAV has been a virus that's evolved over time to transduce human cells efficiently and does so efficiently, non-viral technologies such as lipid nanoparticles really don't have the capabilities currently to be able to efficiently get DNA payloads into the nucleus. So moving to the different classes of in vivo DNA delivery vehicles, there are obviously viral particles, non-viral particles, where viral particles have been efficient in the clinic, such as AAV, but have shown significant limitations when it comes to cargo capacity. Its immune profile is bad in that it doesn't allow for the redosing of the therapy. And then manufacturing of large doses is very tough, given it has to be done in cell culture. And then non-viral particles largely solve a lot of those limitations, but have had a translation problem when it comes to the efficiency. And we largely believe that's because DNA is a payload that needs to get to the nucleus and not the cytoplasm, which are most LNPs are designed for. At Genentech, we're pioneering this genetic encoded nanoparticle, which we believe gets the best of both worlds in having large cargo capacity, low immunogenicity, and manufacturing at scale. But we have a protein shell that is genetically encoded that we can connect to processes such as high throughput screening and directed evolution to evolve for those features. And in addition to that, we'd like to highlight that our particle is derived from a member of the microbiome or commensal flora. And since birth, we do have a very significant exposure to phages on a daily basis, where it's been estimated that over 30 billion phages actually transcytose from our gut into our circulation daily. In addition to that, phages have been used in anti-infectious indications, and there's over a decade of clinical human experience showing that redosing is safe for those therapeutics. So the foundations of our technology lie in the PDP capsid protein, which are shown here in the red, orange, and turquoise colors. Those three colors shown are the three different capsid proteins that we can engineer. They can be engineered in a modular way, and importantly, they're genetically encoded, so we can actually equip them and evolve them in the same ways that nature evolves viruses. Inside this capsid, there's a minimal phage DNA or MIP DNA that has pretty attractive features when it comes to gene delivery, and that its cargo capacity in our hands is over 20 kilobases, and interestingly has a covalently closed single-strand DNA topology. And we believe this also may have advantages when it comes to not integrating into the human genome. The process of discovery for our PDPs is really focused on our Forge platform for in vivo delivery. So this Forge platform means functional optimization by RNA-guided evolution. And the three pillars of this platform are Forge manufacture. So that's the ability, the in-house know-how to be able to manufacture our PDP variants with high purity and high scale. Our Forge screen platform, which really is the evolution process to find that needle in the haystack of a variant that has a target biodistribution of interest. And lastly, our Forge machine learning pillar is the analysis of all the high throughput screening data that ultimately enables us to iteratively design better and better PDP variants for gene delivery. So to touch on our Forge manufacturing platform, there are two main ways in which we can actually functionalize our PDPs. On the left-hand side, we can functionalize it through genetic functionalization. So this is classically known as phage display. This has the ability to display moieties such as antibody fragments, cell-penetrating peptides, endosomal peptides, and nuclear transpeptides. And in the case that our moiety actually is not genetically encoded, we also have the ability to perform chemical functionalization, where we can conjugate things such as polymers, lipids, and molecules to our PDP coat. So an example of what Forge manufacture enables us to do is starting on the left-hand side, where we have a wild type phage particle with no functionalization. We can use our Forge manufacturing platform to equip the three capsid proteins with a targeting moiety that enables successful cell targeting. And what we see in some of our early in vitro data is that this functionalization is key for transduction. So in the first column of images, the top shows the internalization profile of a non-targeted PDP. You can notice that there's very few magenta colors inside these cells, but the moment that we functionalize it with a moiety known as RGD4C, shown in the bottom left, that these PDPs get into cells very efficiently. And this translates over to gene expression. If we actually encode a GFP transgene within our PDPs, you can see that the non-targeted PDP does not facilitate transduction, whereas the targeted PDP on the bottom right does. We have moved those studies in vivo just to see if the profile is specific, and using, again, a known RGD4C targeting moiety and delivering an IL-15 mRNA, IL-15 gene cargo. We use RT-qPCR to basically measure the expression of the IL-15 mRNA across diverse tissues of the mouse. And RGD4C is primarily a tumor targeting agent. And what we can see here is our PDPs in vivo can drive very, very specific expression towards the target tissue of interest, in this case being the tumor. So that's a proof of concept when it comes to using our Forge manufacturing platform and showing that we can get indeed cellular transduction and a specific biodistribution in vivo. But our ultimate goal is to actually target non-tumor samples such as the heart, the muscle, and the CNS. And Forge Screen really is that process for us to be able to do so. So starting on the left-hand side, we're really using the foundations of phage display to equip our phage-derived particle at 3 different points of our capsid protein, in which we can perform modular evolution across all these capsids. On the right-hand side, describes on a high level our process. We construct billions of these PDP variants in a pooled fashion. And then we have a proprietary Forge screening platform that we do in vitro and in vivo evolution studies to be able to find those particular PDP variants that biodistribute in the way that we want in organisms such as initially mouse and non-human primates. In terms of our Forge Screen enhanced evolution process, we'd like to highlight there are really strong differences in what we do compared to traditional phage display. Our Forge Screen is really developed to evolve towards enhanced transduction and not just target binding. So traditional phage display here in turquoise, shown here in navy blue, really selects for phages that like to bind to the surfaces of cells and maybe get into the cell initially, but don't lead to productive transduction events. We've really built our Forge Screen platform to have more downstream readouts, such as a DNA readout inside the nucleus of cells or an RNA readout following the successful transduction of the cell to be able to select for variants in a more informative way. Moving to the last pillar of our platform, these screens that we're performing generate large amounts of data. So we use very deep next generation sequencing to see our billions of PDP variants. For each one of these variants, we're able to measure multiple things, including the manufacturability of a variant, in addition to the variant's ability to transduce. And we're working on a variety of different machine learning algorithms to be able to understand that information. The ultimate goal of being able to potentially predict downstream the manufacturability of our variants or the transducibility of our variants in an in silico manner. How our Forge ML pillar plays into our directed evolution process is we usually start with screens on the left-hand side that are very broad in variation and diversity. And this is just a schematic that shows that we sample from the theoretical sequence space in a way that is not really focused. But then following our machine learning, what we hope to do is going to the right-hand side, identifying certain features of the ones that have shown really attractive attributes, and then designing from those initial variants to newer variants that may have enhanced properties. So with that, to summarize, we believe that our PDP technology and our Forge platform gives us the ability to potentially access a very significant opportunity for in vivo gene delivery. There is still a tremendous clinical need for delivering DNA payloads outside the liver, delivering payloads also that exceed the cargo capacities of AAVs, and the potential for redosing in the case of a genomic medicine could be a real game changer. And lastly, where we recently raised our seed and have been in the process really pioneering our platform and making sure that we have that engine for PDP discovery. We are in the process of thinking about financing down the road to really bring our platform to an IND. And our hope with our technology is to eventually go beyond rare monogenic diseases to provide a more accessible genomic medicines. And with that, I'd like to thank the organizers of MassBio and all of you who listened and appreciate it.
Thank you, Jonathan. That was fantastic. I also want to give a shout out to our partners, Lab Center Ignite, Rachel, who used to be at MassBio herself, for the wonderful introduction to GenSci. And as far as questions go, what challenges have you faced as a company getting off the ground? and how has it been like for you?
Yeah, so starting from the very beginning, I think one of the challenges that young companies face is definitely lab space. So being, it's a very concentrated talent pool. We wanted to put the company in a position to be able to do the work very efficiently. And there's a lot of incubators in the area, but lab space can be limited. We were very fortunate enough to be able to land in a spot at Lab Central, where through their various offerings, like or through partnerships with pharma like Golden Ticket, we were able to grab some of those and initially get a private suite. That overcoming that challenge has really gone the end journey in terms of like our ability to do the science. The second thing, in terms of our technology specific, it's really that this is a very new technology, and while there are a lot of experts when it comes to phage engineering or in vivo DNA delivery, we're kind of at that intersection, and a lot of the assays that we're developing are a little bit niche, like very focused on the vision that we have, and a lot of that has enabled us to think creatively about our problem, but also has been a difficulty when it comes to trying to figure out who to talk to. But we have reached out to many key opinion leaders, and through the aggregate of information, we find I think we're in a great position to kind of execute on our vision.
So you mentioned that you had to sign a lot of agreements with tech transfer offices and get a lot of licenses. Were there any challenges related to that you'd like to speak to?
Yeah, so that's probably a better question for our CEO, Labi. He primarily led those developments. There's, of course, challenges, as everybody knows. Yeah. But I think all parties are really focused on, getting the value out of the technology, and both sides ultimately are going to get to a place where we think we're doing the best, we're using the patents in the best way, and it's a fair agreement.
What does your ideal collaboration look like?
Yeah, so some of those are, while I can't disclose, are in diligence, but an ideal collaboration that we see is someone with in-depth indication specific knowledge. Our company is extremely technology focused, and that just comes with the nature of where our technology is at. We believe that our technology expands some of the capabilities of in vivo gene delivery. So we want to kind of push some of those boundaries in terms of thinking about clinical indications that maybe weren't previously thought of because of patients, let's say AAV, like maybe you can't redose or the cargo capacity is not as large. So, and finding those indication experts out there who really know what to look for when it comes to designing a preclinical development path. We've been fortunate in finding a lot of those for a lot of indications that may be of interest to us. But yeah, that's something that we look for in a collaborator.
Thank you. And thank you very much, Jonathan. That was a wonderful presentation. We'll be moving on to our next one now.
Okay.
So our next company deals with mitochondria. As all of you know, mitochondria are the powerhouse of cells, and any kind of dysfunction with mitochondria can really be disruptive for anybody that has that disruption or is born with that disruption. So this wonderful company, MitoChem Therapeutics, seeks to address these issues. They come to us all the way from Charleston, South Carolina, through our sister organization, South Carolina Bio. And it is my pleasure to introduce Mike Wowodzki, the CEO of MitoChem Therapeutics. On to you, Mike.
Great, thank you very much. It's a real pleasure to be here. I'd like to thank the hosts at MassBio for inviting us to be part of the First Innovation Week. It's a real pleasure, and it's an honor to be amongst the other presenters today. So as mentioned, I'm CEO of MitoChem Therapeutics. Let me see if I can share my screen here now. All right. So our company is a preclinical stage company developing mitochondrial therapeutics for rare and challenging diseases. The company was founded by a neuroscientist and mitochondria experts at the Medical University, South Carolina, in the Beeson and Rohr labs. And there was the insight that there's a metabolic crisis that takes place early in disease pathology that ends up driving pathology. And if we could treat that disease, we'd have a chance to actually slow or stop progression of disease. We have a next generation approach for in treating mitochondrial therapeutics. They're small molecules that treat a hallmark mechanism of aging called mitochondrial dysfunction. A little over $4 million in disease and science-based experts from places like Foundation Fighting Blindness and NIH have led to a robust global patent portfolio over the compounds and a lead program in a rare degenerative eye disease. So mitochondrial dysfunction sits on the front line of disease response. And as just mentioned, everybody knows that mitochondria are the powerhouse of the cell. Oh goodness. Well, running into my first problem here. There we go. All right, so they produce the vast majority of energy necessary for cell function and survival. They manage the response to oxidative and metabolic stress, as well as calcium homeostasis. They respond to increasing energy requirements by going through biogenesis. They remodel, regenerate, and repair themselves and keep the mitochondrial network in healthy position to provide all the necessary energy to perform their function. They facilitate mitophagy, and importantly, they also regulate orderly cell death through apoptosis. So mitochondrial dysfunction occurs early in diseases I'd mentioned, oftentimes even before disease manifests. So things like gene mutations, aging, environment exposure lead to this metabolic crisis, and if unresolved, it leads to cell death. Our portfolio really is focused on diseases that have mitochondrial dysfunction that begin early and drive disease pathology. And this is all the diseases that remain out there that are really challenging diseases and difficult, and have challenged us for a long time. We think that addressing mitochondrial dysfunction will really give us an avenue to go after these at some point. So our lead program is in retinal degeneration. So it turns out that the eye is one of the highest energy consuming systems in the entire human body. There are lots of mitochondria in all the various layers of the retina. What you see here to the right are a graphic of photoreceptors. On the left are rods, which see light, and on the right are cones, which see color. And aging and genetic mutations in environment disrupt mitochondrial dysfunction. They impair the energy capacity. They create that metabolic crisis that disrupts the regeneration repair process and reduces the ability to respond to stress. And if unresolved, results in cell death. And it's very clear that mitochondrial dysfunction is implicated in the underlying pathology for tissue damage in retinitis pigmentosa, dry AMD, glaucoma, and other key retinal degenerative diseases. This is just a representative example of the literature that's out there talking about mitochondrial dysfunction in these diseases. These are really large markets with unmet needs. Today, dry age-related macular degeneration has over 160 million people worldwide that have no treatment today. And just as a matter of perspective, dry AMD is about 85 to 90% of all macular degeneration. The other 10 to 15% is wet, and wet represents between $9 and $12 billion global market. Glaucoma, while it has intraocular pressure treatment drugs today, there's still ongoing degeneration that persists in the retina. And then finally, retinitis pigmentosa is an orphan disease with no approved treatments today. And if left untreated, ultimately ends up in blindness. So the MitoChem solution are small molecules that are designed to restore the energy capacity, to slow the damage, and to improve potential for repair. Our lead product candidate is called MC16, and it's been down selected because it's optimized for topical ocular delivery. And our first indication is going to be retinitis pigmentosa. We've got preclinical studies that demonstrate efficacy in both RP and AMD. We've got early favorable safety and tox data. And importantly, MC16 for this specific disease has the potential to work in all genetic forms. So RP actually is an accumulation of hundreds of genetic mutations, all underneath the umbrella of retinitis pigmentosa. And so the elegance of this approach is, unlike a gene therapy that targets a specific gene, MC16 has the potential to protect regardless of the underlying driver. We have a number of preclinical models and proof of concept that demonstrate efficacy moving forward. I'm going to talk about two of them. I'll talk next about the RPE serum starvation model. It's a model for AMD that shows that stress cells with MC16 increase mitochondrial capacity. And then I'll talk about the RD10 mouse model, which is a genetic model for RP that demonstrates both preservation of structure of photoreceptors as well as improvement in function in cone photoreceptors. So let's start with the mitochondria and their capacity. So MC16 increases mitochondrial function in stressed retina cells. So the retina is composed of a number of layers of cells. One important one is called the RPE. And the RPE is the blood-brain barrier for photoreceptors and is vital for their function and survival. Mitochondrial damage in the RPE occurs early, just like I said before, and aging impairs RPE cells' mitochondrial capacity. So the graph on the right represents output from the Seahorse respiration instrument. It's an assay that, if you're not familiar with it, think about this as a treadmill that you put mitochondria on to determine their ability to produce energy. And you can measure their resting rate or their baseline rate, and then you can have an uncoupling response that forces mitochondria to maximum output. And so in this assay, we grow RPE cells in a serum-starved environment as a proxy for aged stress cells that you'd find in AMD. And what we find is that pre-treatment with MC16 dramatically improves the energy capacity of mitochondria, not only in the baseline, but importantly in their maximum response, which is exactly what we want to see. Next, I'm going to talk about a mouse model called the RP. It's the phosphodiesterase E6 beta mouse model. It's a phenotypic copy of what's found in humans. And in this slide, we're counting rows of photoreceptors. So after treating mice with MC16 for a period of 10 days in this very aggressive model, we then count photoreceptors in both the dorsal or the upper retina, as well as the ventral or the lower retina. And what we see is that MC16 protects a significantly larger number of rows of photoreceptors in both the ventral and the dorsal retina. And you can see here on the right, the histology, just giving you a visual representation of the difference in number of rows of photoreceptors preserved with MC16. And this is clinically important because the loss of photoreceptors is a direct correlation with function and visual field sensitivity in patients with RP. And then here in this data set, I'm showing that MC16 improves the visual response of cones in this model. And so this is really important because as human beings, we rely on cones for our central field of view, and it's critical to our vision. And what we see, not only are we able to preserve the structure of photoreceptors, but in this model, we can demonstrate an improvement, roughly between 30 and 60% improvement in cone response, which again is exactly what we want to see for vision in humans. So we continue to work on trying to understand what is our mechanism of action specifically in our target. And based on Western blot pull downs, we believe one of our targets to be a protein called fused in sarcoma, or FUS, and it predominantly resides in the nucleus, where it enhances gene expression and processing related to bioenergetic regulation, mitochondrial homeostasis, and antioxidant response. And under stress, this protein translocates into the cytoplasm and ends up in aggregates called stress granules and around mitochondria. And if unresolved, what ends up happening is the mitochondria are forced out of homeostasis, and they tip the balance to fission, and excess fission leads to an unwinding of the mitochondrial network, which ultimately leads to cell death. And so through internal studies and testing, we've demonstrated that MC16 will actually inhibit the aggregation of FUS at mitochondria and stress granules. And in a post-treatment environment, if you have aggregation around mitochondria, MC16 will actually resolve that aggregation. And we think that this is meaningful. So our development pipeline consists of major eye and kidney indications. We are farthest along with retinitis pigmentosa in IND enabling stage, dry AMD in preclinical, as well as glaucoma. And we've made some early gains in nephrology, another high energy system for both an acute kidney injury model and diabetic kidney disease. So let's just talk about competitive advantages at Mitochem. So our gene mutation agnostic approach to protection is truly unique. It's not an antioxidant. This is a next generation approach that isn't challenged by the fact that you have to have a lot of antioxidants on board continually to deal with the reactive oxygen species. Our compounds actually upregulate biogenesis and antioxidant pathways. They're gene mutation agnostic, so whereas gene therapies target a specific gene, we expect our compounds to work regardless of the genetic mutation, and we think that that's really meaningful. These are really complicated diseases, and we expect that the final treatments for any one of these diseases are going to be multimodality, and MC16 and its ability to treat mitochondrial dysfunction we see as fitting right into that multimodality approach. As a target that goes after mitochondrial dysfunction, we believe that gives us a platform for being able to take our compounds not only to multiple indications within the eye, but for those indications outside the eye. And importantly, given that most RP patients are diagnosed in childhood, we have an ocular drop delivery, and we think what we see as superior over a lifelong treatment of injections into the eye. And then lastly, all of this is protected by an exclusive global IP portfolio with over 30 patents issued. So our milestones completed to date: we have a lead molecule selected, in vivo and in vitro proof of concept models done, biodistribution, early safety data, as well as FDA orphan drug designation for retinitis pigmentosa. And just this past summer, we raised another $1.5 million in equity to continue our development. So our next milestones all lead us to clinical trial with an IND allowance. So with $5.5 million as our target raise, we would complete our pre-IND meeting with the FDA, our IND enabling studies, our GMP scale-up and production, as well as completing our phase 2 clinical design with the ultimate milestone of receiving our IND allowance. The current team brings the necessary experience both in management and science. My background has been in science and technology-heavy industries. Started a company previously called Salutaris Medical Devices in the space of radiation ophthalmology for treating wet macular degeneration with therapeutic radiation. Our co-founder, Barb Rohrer, is a neuroscientist and our chief scientific officer. She was the insight genius that started the company, along with a former co-founder, Craig Beeson. Rick Schnellman is another co-founder of the company, bringing pharmacology and mitochondria expertise. He's the Dean of College of Pharmacy at the University of Arizona. Dr. Abner Ingerman is on our clinical and advisory board. He's got a long career in pharma as an MD ophthalmologist, but most notably former head of ophthalmology over the Regeneron program, over the EYLEA program at Regeneron. And then lastly, Dan Adams has joined our board. He's also had a long career within the industry and was one of the co-founders and first CEOs of Biogen. So in summary, mitochondrial dysfunction is an early and significant driver pathology in the eye, and we believe a key target for being able to develop therapeutics that can stop or slow the progression of disease. It's a unique approach to mitochondrial protection with demonstrated preservation of both structure and function. We got key clinical milestones completed to date. The FDA has granted us orphan drug designation for our lead program. We got the experience, the science, and the management team. And importantly, an opportunity to create multiple value streams by funding the RP-IND program while partnering with others, on other indications with companies outside of Mitochem. And with that, I thank you, and I'd like to take any questions that anyone has.
Mike, that was wonderful. Glad to hear what you're doing, and you have a wonderful platform. What's next for Mitochem in terms of partnerships, and what are you looking for? Who do you want to meet?
Well, we've got a number of conversations going on right now, which I can't disclose, but we're looking for partners who are looking at any of the diseases, both within ophthalmology and outside. Given the work that we've done in nephrology, it demonstrates the concept of going after mitochondrial dysfunction. So we've got discussions within companies interested in kidney, as well as companies who are looking at CNS. So we're really looking for partners that have the expertise that we don't have, because our expertise really relies in the eye. So we're looking for those partners that can bring the expertise as well as the resources necessary to advance the compounds.
Thank you. And are you in Boston a lot, or do you?
Well, in the last couple of years, there hasn't been much traveling, but I was back for a reunion recently, and like everybody else, I think on the planet, just really, really dying to get back out and get back into the world. So looking forward to being in the Boston area, given its prominence.
Yeah, well, we do have a lot of great events coming up, and we have our State of Possible conference next year. And you can certainly get lots of great networking going and meet a lot of investors and potential partners. So we hope you can do that. Thank you. We don't really have other questions that have come in. I'm just waiting. There's a little bit of delay on our platform. But in the meantime, what challenges did you have when you put this company up?
So like every company, we have our challenges. When I joined the company just about 3 years ago, and a few months after I joined, one of our two founders, Craig Beeson, who was the mitochondrial expert and the medicinal chemist, was diagnosed with terminal cancer and was gone in 10 days. And so we lost one of our key founders right off the front. And then of course we had COVID, to start off with. I would say one of the challenges is, the great news is that there seems to be a real resurgence in the interest in mitochondria and mitochondrial dysfunction, and a lot of companies focusing on the hallmarks of aging.
Yeah.
And so I think, as we look at that, companies like Merck look at mitochondrial dysfunction as one of several mechanisms that they're looking across disease. And so despite the challenges, knowing that mitochondrial dysfunction and some of these diseases have renewed interest is really encouraging and making all the effort worthwhile.
Thank you so much, Mike. And next up, a quick message from our sponsors. Stick around for the last couple of pitches. We have a couple of really good companies coming up, but before that, we'll move on to a message from our sponsors.
Great. Thank you very much.
and I look forward to staying in touch. Hi, everyone. We're back. Thank you for your patience. Now we have a slight change in the program. We are going to be presenting Brent Stead from Specific Biologics, a gene editing company first, and then we'll have Lizard Bio after Specific Biologics. Brent, you're on. Go ahead with your presentation.
Wonderful. Well, thank you so much. I do truly appreciate the opportunity to present Specific Biologics. I am Brent Stead, a co-founder and CEO here at Specific Biologics, where we are developing a new gene editing technology. Now, if I were to survey everybody on this broadcast, a majority of individuals would have a loved one or someone close to them who has a serious genetic disease. Gene editing technologies are designed to be molecular tools that can go into a cell and correct a disease at its source, a change in the DNA. And that's precisely what we're trying to do at Specific Biologics, where we are developing therapeutics to treat or cure disease through precision gene editing. And we highlight that word precision in our mission statement because our gene editor, Dulase, is a new gene editing platform that has a unique two-site mechanism of action. And it's that two-site mechanism of action that allows us to accurately disrupt or increase the frequency of repair of disease-causing mutations through two distinct mechanisms of action. Now, all this work started in the labs of Dr. David Edgell at the University of Western Ontario, a few hours outside of Toronto, where he first described the activity of these RNA-guided two-site nucleases. We spun the technology out of the university into Specific Biologics and began to develop the versions of Dulase that we work with today. We are proud residents of JLabs at Toronto, here in the Mars Discovery District, where we've established our own labs and generated a lot of the foundational intellectual property that forms our Dulase patent portfolio. We are also graduates of competitive accelerator and incubator programs like Creative Destruction Labs and the California Life Science Institute. Late last year, we completed a seed round financing with leading life science investors, Lumira Ventures and Admare Bioinnovations. And that really brings us to the past year where we have completed preclinical studies that really demonstrate Dulase's utility across a broad and diverse set of genetic conditions. Now, I'll give a little background on why a tool like Dulase is required. There are hundreds of thousands of disease-causing mutations that come in quite a variety of different types. They may be single letter changes in the DNA or large and small insertions and deletions. With current gene editing technologies, the cell repair pathways that are used are often unpredictable or inefficient, and that really limits the therapeutic application of a lot of these gene editors. And that's where Dulase comes in. Dulase is a single-chain two-site nuclease, or gene editor, in other words. It is comprised of an engineered version of a Staph aureus Cas9 domain that is guided to its target site by RNA. That Staph aureus Cas9 domain is fused through a linker to a second nuclease domain called ITEV1. And that ITEV1 domain binds and cuts at a 5-letter sequence, as shown in the target site below, that starts with a C and ends with a G. But it will not cut that target sequence unless it's bound to the right site by that Cas domain and that ITEV1 site is properly spaced from the Cas site. And so we have a nuclease or a gene editor that has really high specificity for a target site that can cut DNA in 2 spots, hence the name Dulase. And so it's interesting that it cuts in 2 spots, but what's really important is that when it does cut, it leaves unique DNA ends. And it's really those unique DNA ends that allow us to exploit the preferred repair pathway in cells in order to generate these defined length deletions and increase the frequency of repair. So when Dulase binds to its target site, as shown on the left, it can cut twice and remove a fragment of DNA. And at the ITEV1 site, you have a 2-letter overhang, and at the Cas site, you have a blunt end. And so it's these non-compatible DNA ends that we can exploit in order to push the cell to create defined length deletions or increase repair. Now, let me take you a layer deeper into some of the data to support those two distinct mechanisms of action. So in the disruption mechanism, the two ends that are cut by Dulase can eventually meet. And through the preferred cell repair pathway, the non-homologous end joining pathway, you can end up with a defined length deletion between those two sites. And that's precisely what we see in cells that we treat with Dulase. So shown here in panel B are cells that are treated with Cas, the single enzyme CRISPR-Cas, and we sequence the nature of the editing outcome by deep sequencing. And what we see is what everybody else sees, those random insertions and deletions around the cut site. But with Dulase or TEV-Cas in panel A, you see that bias for that defined length deletion. And that's what got us really excited about this technology, is the ability to generate these precise length deletions. And then we took it one step further and asked whether or not that we could increase the frequency of repair because we end up with these non-compatible ends. So this is a similar setup where we treat cells with Dulase, but we also include a repair template of DNA that has the same ends that we've cut with Dulase, an overhang and a blunt end. And again, through the preferred cell repair pathway, the non-homologous end joining pathway, the cell can paste that sequence into those cut sites. And that's what we see inside of cells, this high frequency insertion of that repair template into the cut site. And in fact, when we look at the levels of repair, they're near equal to the levels of editing. And that's because this non-homologous end joining pathway is a preferred pathway. And if we stack that up against levels of repair, we can see with homology-directed repair, the non-preferred repair pathway with Cas, our pathway is an order of magnitude different. And it's really important because that pathway is active throughout the cell cycle and available in most cell types, which really unlocks a number of applications that we can pursue. And of course, with any gene editor, precision for the target site is paramount. The gene editor has to bind and cut at only the on-target sequence and no potential off-target sequences within the genome. And that's precisely what we observe across the target sites we've tested. And this is just some data that does support that. And I'll just set this up where you can see on the left-hand side, the on-target sequence with the Cas site and the TEV site, and we predict within the genome other off-target sites where there's little mismatches denoted by the stars. And sometimes even where there's a properly spaced ITEV1 site from those mismatches. And the takeaway message here is that if we look at the editing, we do not detect any editing at those off targets, both through methods like this gels as shown here or through sequencing, which supports the precision of the Dulase platform. And very importantly, as a platform, we need to have robust editing across many clinically relevant targets and cell types. And that is what we observed. We've edited multiple different clinically relevant targets and multiple different cell types. And we observe that Dulase has robust editing efficiency across these targets and cell types, showing that it has a breadth of a platform. And very importantly is getting Dulase to the target cells. And fortunately, Dulase is compatible with the available delivery modalities in order to target quite a number of cell types. We have been developing proprietary application-specific lipid nanoparticles for delivery, but our technology is also compatible with preferred delivery methods like adeno-associated virus, because the coding sequence of adeno-associated virus is less than 5 kilobases, and our coding sequence of Dulase is only 3.7 kilobases. And so we fit within the packaging capacities for an all-in-one adeno-associated virus for applications where we need to use that delivery vehicle. And we are actively exploring other partnership opportunities and other delivery modalities inside of these. And so Dulase is designed to deliver the right edits, but also with maximum flexibility. When we stack ourselves up against early versions of gene editors, as well as emerging versions, we can really differentiate ourselves on having precision disruption and increased frequency of repair across a broad range of targets and cell types. But very importantly, DUALASE is designed to be a good gene editor in that it is small to fit into coding sequences of adeno-associated virus, highly programmable through the RNA-guided domain, and it has higher specificity for its target site through the engineered variant that we use, as well as the requirement for 2 sites. And what that allows us to do is explore unique applications where our 2-site mechanism of action has a differentiated target site because of a disease-causing mutation. So let me just walk you through a couple examples here. You can imagine in a gene where you may have small deletions or large deletions of nucleotides within a target sequence that you want to either disrupt or repair. Those deletions can oftentimes form a site for DUALASE that doesn't exist in the normal copy of the gene. And so we have the opportunity to just target the mutated allele for either disruption or correction because of the creation of those target sites. And the same holds true for small and large insertions, and even base changes that aren't served well with base editors that are available today. All this to say that there is a set of mutations that are not easily targetable with current technologies, and DUALASE unlocks a number of these applications and targets. And really what we've done is use bioinformatic prediction to look at a large number of clinical target sets where we would provide a unique advantage with our 2-site mechanism of action. And that really allowed us to narrow down on some potential clinical applications that we would like to pursue and have pursued. And so we have undertaken a set of studies and completed a set of studies in a broad range of different targets in liver, ocular, and central nervous system disorders. This is a set of 6 new targets that we've been exploring within the last year for both the gene correction and gene disruption application. We were also fortunate recipients of a grant from the U.S. Cystic Fibrosis Foundation to support some of our early work in the cystic fibrosis area. And I will actually take you through a couple data points in that because those are the publicly disclosed targets that we've been pursuing. But we also have other funded programs in engineered cell therapy and are proud members of the Blue Knight collaboration, where we are trying to deploy some of our lipid nanoparticle technologies for pandemic preparedness. So let me just quickly walk you through an example of how DUALASE works within cystic fibrosis, and what you can do is imagine how this could be applicable in other disease areas that we've been exploring. And so our strategy here is try to correct the disease-causing mutation in CF patients in the lung, which are mutations in the CFTR gene. There are available therapeutics for some of these mutations, but oftentimes patients have things that are called nonsense mutations, where there are no available therapeutics. And we are targeting a set of these mutations. And just to show you our strategy again, what we are doing is actually cutting on either side of the mutated sequence with our DUALASE technology, where we can insert a correcting sequence into those 2 cut sites. And we have been developing nebulizable lipid nanoparticle formulations to deliver our gene editor to the cells of the lung. And so when we look at the activity of DUALASE on sets of CFTR mutations in epithelial cells, as well as in vitro, we first observed that DUALASE is specific for the mutated sequence. That's where we have that preference for the mutated allele. That we can deliver our DUALASE gene editor into epithelial cells through lipid nanoparticles. And importantly, that the levels of repair that we observe inside of those cells with DUALASE are near equal to the levels of editing and above levels that are thought to provide a therapeutic benefit to patients. And in fact, we've actually translated this into functional correction in CFTR mutant cells. So just to set this up, this is a Western blot of cells that we've treated with either DUALASE or with Cas only as a comparator. The top band, or band C, is a full-length CFTR wild-type protein. And these cells harbor a truncation mutation, which cannot produce full-length CFTR. So the presence of this upper band or C band indicates that we're producing wild type cystic fibrosis membrane protein. And that's exactly what we observe with our dual-A's constructs. The corrected cells contain functional copies of CFTR, or wild type protein, by Western blotting, thereby showing that correlation between correction at the genotypic level and functional correction inside of the cell model. And so with that data, I just would like to spend a moment to talk about the team behind the company as well. Myself, I'm co-founder and CEO. Prior to this, spent a number of years in industry, as well as completing my doctorate at the university where the technology originated. But very importantly, coupled with a co-founder who comes with years of experience in operations and process development, and very importantly, expert advisors. We have independent board members who have years of experience in the pharmaceutical industry, as well as domain expertise in the gene editing field. The inventor of the technology, Dr. David Edgell, on our scientific advisory board, as well as subject matter experts like Dr. Amy Wong in the cystic fibrosis programs. And we are very fortunate to be funded by world-class life science investors like Lumira Ventures and Admare Bioinnovations. And then last, I just want to leave it on that we do have a robust intellectual property strategy in place to protect our dual-A's platform, including entering the national phase with foundational application that we filed a number of years ago with office action expected shortly, and are actively generating an additional IP to strengthen that portfolio, as well as differentiating ourselves as a dual site or two-site mechanism from existing gene editing landscape. So with that, I'd like to leave you on three thoughts. First is that we do have a robust data package across a variety of disease-causing mutations that demonstrate the breadth of dual-A's platform as an efficient and precise gene editor. We've also built a pipeline of clinically important therapeutic targets on which we're now nominating a number of development candidates. And last, I think, is to leave you the message of hope that these types of technologies represent an extremely important scientific advance, where we can start to talk about the potential for gene editing to treat or cure diseases at their source, a change in the DNA. And with that, I'd love to answer any questions that may have come up throughout my presentation, and thank you for the time.
Thank you, Brent. We do have a question coming up from the audience. What are some of the key challenges in terms of gene editing? And how large of a fragment can the database dual-A's based gene editing achieve with a single gRNA?
Perfect. So yeah, so I'll answer the second one first, and then I'll come back to the challenges and opportunities, I'll call them. So the short answer for the target site for dual-A's and how much of a fragment can we cut, it ranges from about 30 to 36 base pairs. However, we can insert much larger sequences than that, and have shown that we can insert much larger sequences than that. The challenges and opportunities with gene editing, I think the challenges are often around delivery. We hear that quite often about getting the gene editor to the target tissue. And I think that's why you really have to take a pragmatic approach of looking at, and one of the reasons why our pipeline is where it is initially, are tissues we think we can target with today's technologies, while we develop new versions that can target new tissues. And so that's why we've really narrowed down on that set of targets initially. And then the other two challenges that we really do address with our dual-A's technology is precision, making sure that it only cuts the target site on target, and efficiency. It has to get above the clinically relevant thresholds of gene editing.
Thank you. That's
wonderful. Thank you so much. I really appreciate it.
Great having you here. Thank you for joining us all the way from Toronto.
My pleasure. Thank you so much.
Yeah. And now we are going to move on to our final presentation of the day, Lazar Bio. They are coming here all the way from Brazil. I don't know where you are right now, Marcos. But Marcos Valadares, the CEO of Lazar Bio. And they also come to us by way of our partners at Lab Central Ignite. Thank you so much, and thank you for joining us today.
Thank you, Gary. It's a pleasure to be here. I would like to thank the team that invited us as well, and by giving us this opportunity to share what we've been working on. My name is, as you said, Marcos Valderes. I'm the CEO and co-founder of Lizard Biotherapeutics. We do have Brazilian operations, but we also are operating in the US as well. And glad to be joining you here today. Let me share my screen. Let me put this in presentation mode. There you go. I'm assuming my presentation is being seen right now. Am I correct?
Yes.
Okay, thank you for that. Appreciate it. So this is Lizard Biotherapeutics, and we are a cell therapy company. We want to cure incurable diseases using cells. We do believe cell therapy holds great potential to improve human health, and we understand there is a manufacturing challenge around this. And therefore, at Lizard Bio, our main goal is to tackle, to industrialize cell manufacturing for cell therapy applications. And I'm going to start off by showing a very large problem around heart failure. Every 42 seconds, someone has a heart attack just in the US. Fortunately, more than half of these people are surviving this event. They survive the event, but they go on to develop a chronic condition known as heart failure. Over 6 million Americans have been diagnosed with this already, which adds a huge pressure on the healthcare system by accounting for more than $44 billion in medical expenses every year. And the disease itself is simple to understand. People lost heart cells at some point of their lives, but they're never able to get them back, at least naturally. But this is where we come in, because for the first time in history, we are able to manufacture bona fide human heart cells at industrial scale and cryopreserve them for future off-the-shelf use. And this is something that's really exciting to us. And we've been able to show that these cells, they beat by themselves, they beat in synchrony, and therefore show all the features necessary for a heart cell. And I'm also going to dive a little bit deeper on this in vivo data that I'm showing here this year, but just to preemptively already say, we've shown these cells can improve myocardial infarcted rats, and we can remuscularize pig's heart with these cells. So all of this is very, very exciting. And we used to say that every time we look at this data, our hearts skip a beat. But how precisely are we actually producing these cells? So we are relying here on a couple of different technologies. So we're talking about using pluripotent stem cells in order for us to generate potentially any cell type in the body. We are talking about using hypoimmunogenic cells in order to avoid immune rejection. Those are not technologies that comes from within Lizard Bio. This is outside technology that we're in licensing. And the last point, this is where we are actually generating value here. Our idea is to create these cells in huge amounts in industrial scale, and this is where we are focusing and where our technology comes from. So let me just dive into the data that supports our claims. We're talking about a technology that is able, our technology is negligible cost media. Basically, the only thing we noticed, we learned from everything that we've done so far, that the only thing that is scaling linearly and doesn't benefit from economy of scale when you're scaling cell therapy is media. So current commercial alternatives are just prohibitively expensive. So we began by developing a negligible cost media that supports pluripotent stem cell cultivation and differentiation in both 2D and 3D. And hopefully I'm able to convince you of that in this presentation. And we do have a proof of concept showing that we can produce as much as 1 billion heart cells derived from pluripotent stem cells, which is roughly the dosage for one patient for less than $20 worth of media for the entire process. And we also have positive in vivo evidence showing that these cells do hold therapeutic value. So first, this is the work of Paul Burge, one of our co-founders. His work is he's working from Northwestern University in Chicago. And there's this publication in which he has shown already a very simplified formula for both a formula of media and simplified manufacturing process of the components to come to a stable and very simple media that is currently less than $1 a liter. And this is compatible with weekend-free scheduling cultivation of pluripotent stem cells, which is generally something very, very difficult for pluripotent stem cell cultivation. And it's patent already, it's patent pending, but we are already in the regional phase of this patent in several regions of the world. And in the end, as I said, we are now at less than $1 a liter. It's at least 100 times less expensive than anything out there. And we do have partners already in place for CGMP versions to be produced, because we are not a media company. We are a cell therapy company. So we are leveraging the knowledge on media to develop a cell therapy, a cardiomyocyte cell therapy base. And this media is also compatible with suspension cultivation. And we've done this in 100 mLs and showing that it is, in average, 5 times better to generate enough cells in 100 ml suspension than we've seen in the literature. But we've also made a step further and started cultivating this as 1 liter suspension systems. So this is just a picture showing that we had 8 systems, 1 liter systems running parallel. Each one of them for the entire process would take around 12 liters of media in total. And this would eventually cost about $48,000 to run that experiment. But we were able to diminish that to less than $300, which is very exciting and gives us a lot of opportunity to develop the process. And this is basically the setup, the differentiation scheme. So you have 5 days of pluripotent stem cell cultivation, another 10 to 12 days of differentiation. And this would be enough to take to generate 1 billion heart cells. And our preliminary data shows that we can do exactly that. So after we inoculate the cells in 1 liter system and until we get by day 20, we're spending less than $20 worth of media, and reproducibility is not where we want to be yet, but those were just the first runs, very first runs we did with the experiment, and we were able to generate up to 1 billion heart cells. So the key takeaways here is that we have validated this with over 100 cell lines. Those cell lines have been both reprogrammed and differentiated and cultivated in this media. Pluripotency is stable. We haven't yet to capture genomic abnormalities, though I have to say we haven't really flipped these cell lines upside down. So there are more in-depth work that we can do, but regular screening, genetic screening is just okay. It doesn't show any problems. No harm in differentiation, simplified composition, 100 times less expensive, chemically defined, xeno-free. And therefore, we think it's a good fit for large scale capabilities to cultivate iPSCs. And we also have generated in vivo data showing that cells, cardiomyocytes that we produce, are able to improve heart function in rats. So this has been published last year, showing that the cells that were injected in rats compared to rats that were non-injected, we were able to improve heart function as measured by left ventricle ejection fraction improvement. And when we compare this to landmark studies such as Laflamme's 2007, we were able to generate twice as many heart cells or human tissue inside graft sites inside of the rat's heart. So this is very exciting and made us move further to pig studies, which we have also generated, though we have a small cohort of pigs. If I'm not wrong, we're talking about 4 animals that were injected. We were able to see, and the protocol was the same. We myocardial infarcted these pigs, and after 1 week, we open their chests, injection into the heart. We're talking about a catheter balloon myocardial infarction. So this is a ischemic reperfusion model, IR model, not closed artery model. So this is ischemic reperfusion, more closely resembles what the patient faces in the clinic. And we've seen a lot of human engraftment, human cells engraftment in pigs. Actually, when we compare this in average, we fared very well compared to large animal studies that have been published in the literature. And this also made us go a step further, or want to go a step further. We can mature these cells. These cells are maturing in vitro. They have blood reperfusion, and it's likely not being rejected when we look at rejection markers for immunological cells inside of the tissue of these animals. So the key takeaways here is that we can improve function in myocardial infarction rats. We can engraft cardiomyocytes in pigs. Rats are twice as better than landmark publication studies. We have competitive data for pigs, and also we have ability and knowledge to run those studies in-house. The team the main work here on cell cultivation is being done by Paul Burridge. The animal work is being done by Rafael Darioli, and the team has been able to accomplish this to develop this further as well. Together, we have over 100 days of experience in stem cell biology and cell cultivation, a lot of experience in vivo work and preclinical tests. We also have been able to get quite significant traction so far. We have some interesting letter of intents from companies, biotech companies in the field that are reputable, scientific partnerships as well with key universities, and also manufacturing partnerships with pharmaceutical companies that have manufacturing, that have a state-of-the-art manufacturing in place already that could be deployed for such a work. So in a nutshell, if I can say, we have exclusive rights, worldwide rights to media and processes that enables us to be the only company actually aiming to cultivate cells at industrial scale. When I say industrial scale, I'm talking about 100 liters or more. We're not at that level yet. I'm going to tell you where we are today, but that's exactly where we are aiming at. And I guess I can say, at last, we're industrializing cell manufacturing. That's my bottom line here. And as for heart failure, we think this is a very relevant market opportunity, aligning the interests of everybody involved. So we have patients that obviously want curative therapies, access to curative therapies. You have pharmaceutical companies that want to have access to transformative medicines that can be protected for longer periods of time. And also you have insurance companies that obviously want to treat patients and not manage them. So we think this aligns, we think this technology kind of checks all the boxes. And if we address as little as 1% of the patient population in the US for heart failure, this already translates into north of $4 billion in annual revenue, which is a sizable opportunity, and we're excited to be working in this. Currently, we have raised up to $2 million in private investments and almost $1 million in grants. And we're now raising $3 million round, our pre-seed round, to do exactly, to 10x our current manufacturing capabilities. We are currently in 1 liter, and we are aiming to reach 10 liter scale, so that's 10x right there. But with the vision to reaching 100 liters later down the line, and therefore truly support the claim of industrial scale manufacturing of cells, of pluripotent stem cell derived therapies. So I guess I can finalize this saying we are a cell therapy company industrializing cell manufacturing. We're 100 times less expensive than anything out there, at least for cardio. We have programs in cardio and potentially can partner for other indications. And I always like to finalize this by saying we are doing for cell therapy and regenerative medicine industry what chip companies have done for the computer industry. They have made a transformational technology accessible. And so if cell therapy is truly the future of regenerative medicine, then we believe that Lizard Bio with its technology is holding the key to make it mainstream. And if you are willing to be a part of this or interested in this in any way, don't hesitate to say so. I'll gladly take any questions now, and I appreciate your time. Thank you.
Thank you, Marcos. That was fantastic. It's so great to end today's program with a company that ticks off both themes for today. cardiovascular and metabolic and rare diseases as well, because I see that you're also working on Rett syndrome. This is fantastic. Tell me, what challenges have you faced as a company in the field? It is not an easy technology yet. You've navigated it. You have this wonderful IP around, scaling up and developing stem cells, pluripotent stem cells.
Yes, thanks for that question. I believe the main challenge now is to change the mindset of the development path, because we are still in the early days for cell therapy. So even though we have some successful cell therapies out there, scaling up is still a second, let's put it this way, second challenge to face. People are still going first for the clinic to see, obviously, if it works. And if it works, and then you go to the commercial phase with whatever process you have, regardless if it is scalable or not. For you to change that before you have the data, if it is efficacious or safe in the clinic, is a really tough challenge, because you have to double down on manufacturing even before you know if you have any potential clinical outcome that is relevant in any way. So this is a change. We believe cell therapy is really going to stay for the long run. That's why we're giving that bold step, let's put it this way. But it's being challenged to find partners and to find people that are willing to take that challenge with us, along with us. So we're pushing forward. We believe that's the way we should go. But it is a change, and we understand why. Obviously, I mean it's there is a logic behind why you don't scale before you know it works. We just believe cell therapy will be available for many diseases and many different patient populations, and therefore pluripotent stem cells is the key to make it scalable, and that's why we're focusing on first pluripotent stem cells, and specifically speaking right now, cell therapy for cardiomyocytes for heart disease.
So I know that from reading about it, not that I have any personal experience doing this, but I know that cardiomyocytes are notoriously difficult to work with, and cardiac cells as well. So how have you navigated that?
Yes, it is true. I mean, there are several challenges after you have the cells, right? I mean, there's safety issues regarding arrhythmias, and also you got to make sure these cells are not being rejected in the end. We just believe that most of these issues, we have some attainable technology to deal with. So when we talk about being rejected, we are really, really hoping here and bringing in the hypoimmunogenic technology. So gene editing to make sure these cells are escaping the immune systems. I think this is not a new technology out there. A lot of companies are going around this, and I think there is a lot of hope behind this. And the science is very strong as well. So we're excited about this. As for the arrhythmia, what we've noticed is in animals, we haven't been able to track our animals yet, but from the literature, the arrhythmias arise and they stay for as much as a month after the injection, but then they subside because apparently the cells are maturing in vivo, and therefore they are not prone to arrhythmias anymore after that. So we're basically dealing with the fact that we need to take care of the patient or arrhythmia cells for a month. And there's some publication already showing that current pharmacological treatments are able to deal with this successfully. So even though we might not have the perfect cell type that does not generate arrhythmia yet, some people are working around this. We think the pharmacological treatments would be able to make a good case for at least a subset of patients, and therefore take this to the next level. And eventually, with more data, clinical data coming in, you would make the case that you can expand this access to other patients because it's now safer than was before. So that's the kind of the path we're going this, but it is a tough path to go, and we're making everything we can to make it as safe as possible, to make the claims based on the data that we have.
Fantastic. And with that, we end our Q&A for today and the program for today. Thank you, Marcos, for joining us, and we're so proud to have you share your presentation on this platform and be a part of it.
I really appreciate the opportunity. Thank you guys for giving us this opportunity to share what we have, and hopefully we'll be around as well to discuss this with other people, other folks that are interested.
Yeah, and I want to thank our audience for being here today. We'll be back tomorrow with some companies showcasing their neuroscience therapeutics and platforms. And we hope you can join. Thank you so much. And we'll move on to a quick message about our sponsors. Thank you, and upcoming events.